Hello everyone, welcome to the ITLS Academy. Today we are going to perform another experiment, that is your gram staining. Okay, so before starting the gram staining, we are going to discuss about the staining. Staining is a microscopic technique which is used to contrast the microscopic image, which means that the image that we get from the microscope, this staining create a easily visualization of that image. Okay, that's why we do a staining. It enhance their color quality, or it enhance the color of that particular bacterial cell. Okay, so we all know that this is known as a staining, and then after staining, we all know that the microorganisms are what? They are a colorless organism. Okay, so if the colorless organisms are visualized under the microscope, then it cannot be visualized. Okay, it cannot be seen with the help of that microscope, or maybe we can see, but the quality of that microscope, the quality of that image are not very good. That's why we do a staining, because it highlight that area of the bacterial cell and also increase the or also add the color into the bacterial cell. That is called a staining. Now the experiment that we are going to perform is a gram staining, which is most common type of the staining and also a most important and widely used technique. Okay, widely used staining techniques in a microbiology. Okay, if we talk about the microbiology, then the most widely, the most common, the most important staining is what? It is a gram staining. It is introduced by a Danish bacteriologist Hans Christian Gram in 1884. That's why the name put it in his name, that is gram staining. Okay, now here what we have to do, we all know that the gram positive, they divide the bacteria into two categories. One is your gram positive bacteria and another one is your gram negative bacteria. Now the gram positive bacteria have a thick peptidoglycan layer. Okay, and these gram negative bacteria have thin peptidoglycan layer. Now when we perform this experiment, or we use a primary stain, that is crystal violet. So gram positive have that thick peptidoglycan layer. They hold that, they make a bond with the crystal violet, and when we wash it, then it will not wash from the distilled water, and also does not decolorize with the help of the ethanol. But in case of the gram negative bacteria, they have very thin peptidoglycan layer, and when we use a decolorizer, then it will decolorize and does not retain any crystal violet color. Okay, now after that we use at the end of at the end of that procedure, we use a safranin. Safranin is a secondary stain. Now gram positive bacteria have already that crystal violet color. Now these gram negative bacteria who have a very thin peptidoglycan layer, they will make a bond with the safranin. Now when we examine these cells under the microscope, the gram positive bacteria shown purple color and gram negative bacteria shown a pink color. Why these bacteria shown these color, that I already told you, that due to that layer of the peptidoglycan. Gram positive have a thick peptidoglycan layer and gram negative bacteria have thin. That's why they will show these color. This is a theory part of that experiment. Now come to the practical part. Now for performing this experiment, first of all we required a crystal violet. This is our crystal violet. It is a primary stain. Then we required a iodine. It is a moderate. Then we required a ethanol. It is what? It is a decolorizer. Then at the end we required a gram safranin. That is your secondary stain. Okay. Now what we have to do? First of all, we sterilize the inoculating loop by a heat that we get from the that burner. After sterilization, it we have to cool down it. Okay, then after cooling, we take it with the help of that inoculating loop. We take out the smear and put it into the glass slide. Okay, then after that, we add a crystal violet. We add crystal violet for a 1 minute. Okay, now when we take out the smear and transfer it into the slide, we heat fix it. After heat fixing it, we add a crystal violet and we leave the crystal violet for a 1 minute. After that, we use our distilled water for a washing purpose, so that the stain will be removed out. Then after that, we add a iodine for a 1 minute. Then again we use a distilled water for a washing. Then we use a ethanol for a 30 seconds. Then again we use the distilled water for a washing purpose. Then after that we add a safranin. Safranin is act as a secondary stain and we leave it for a 1 minute. After 1 minute we wash it with the help of that distilled water, and then we air dry it, and after air dry it we examine it under the microscope. Okay, if the gram positive bacteria are present, then it will appear a purple color, and if the gram negative bacteria are there, then it will appear a pink color. And the at end of the video, I will we will attach the image of the bacterial cell that we shown under the microscope for your better understanding. Now I already sterilized our inoculating loop. Now take out the slide. You have to hold your slide like this or like this. If you hold it like this, then a spot will be appear. Now we have already a culture, that is your pour plate method techniques. We get that culture and how we prepare the media and how we do a serial dilution and how we perform this technique. The videos related to this are already uploaded in our account. You can visit our YouTube channel and see the video for your better understanding. Then after that you will perform that staining. Now what we have to do, we have to take out the smear. See, like this. You can see that smear will be taken out in a loop. Now put it into the middle of that middle of the glass slide. Now you have to heat fix it by passing it from the flame. Now you have to cool down it, and after cool down it, we have to add the crystal violet for a 1 minute. Take a crystal violet. Now we have a crystal violet. We take it out with the help of the dropper and put it into the smear. Cover the whole smear with the help of that crystal violet. Okay. Now we have to wait for a 1 minute. After 1 minute, we wash it with the help of the distilled water. Now after 1 minute, I am going to wash this slide with the help of the distilled water. If it is gram positive bacteria, then it will stain the this crystal violet. Then after that, we have to add this gram iodine into it. Gram iodine is used for the moderate purpose. Okay. Then we have to wait for a 1 minute. Then after 1 minute, we wash it with the help of the distilled water. Then after 1 minute, I am going to wash it with the help of that distilled water. And after that, we have to add this ethanol. Ethanol is what? Ethanol is a decolorizer which decolorize the crystal violet color. And we have to hold it at it only for a 30 second. Okay. So after 30 second, what we have to do, again we wash it with the help of the distilled water, and then at the end of this process we have to add a saffron in. Okay. Now why we always wash with the distilled water? So that if the stain are present, then it will remove. When you are performing the staining, please wear a glove. Now after 30 seconds, we have to wash it with the distilled water. Now after that, we have to add a saffroning as a secondary stain. So if the gram negative bacteria are present, then we all know that they are what? They have a thin peptidoglycan layer, and it will not make a bond with that crystal violet due to their thin peptidoglycan layer. So they will make a bond with this saffronin. Now we will, after 1 minute, we will wash it with the help of that distilled water. Now 1 minute is over. I am going to wash it with the help of the distilled water. Now after that, we have to air dry it, and after air dry it, we have to examine it under the microscope for the see the staining result. If the gram positive bacteria are present, then this cell appear a purple color, and if the gram negative bacteria are present, then this cell show a pink color. Okay, so end of the video we will attach the image of that image that we taken from that microscope for your better understanding. This is all about the gram staining. I hope you will understand, but if you have any query related to this experiment, you are free to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms. That is your Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.